Thank you for inviting me. It's a pleasure to be here today. So the research interest in my lab is age-related macular degeneration. And I'm going to talk about the use of stem cells for this disease as a tool for regeneration, but also as a disease modeling and for development of new therapies. First, a brief review of age-related macular degeneration. AMD is a leading cause of blindness in the developed world. About 11 million people in the US are affected, and in Europe, Europe and US, the number of patients is about 30 million. And with the population, with the aging population, this number is expected to double by 2050. So there is, at this point today, there is no effective treatment for dry form of AMD. There are two forms of AMD, wet and dry. I'm going to explain why we call them like that. And so there is really there is an unmet need for development of clinical need for development of new therapies. The risk factors are aging, of course, but other factors are involved, such as atherosclerosis, obesity, diet, smoking, etc. This is an image seen by a normal eye, and this is an image seen by a patient with AMD. As you can see, the central vision is totally destroyed, and central vision is what we do need to do our daily tasks, such as reading, writing, driving, recognizing faces. So there are studies being done for AMD. First, it's of course now recently stem cells, the possibility of regenerating retina by transplantation. There are some heredity factors that are involved, and there are some susceptibility loci that are contributed to AMD. And there are some form of anti-inflammatory and for the wet form of AMD. But as I mentioned, for dry form of AMD, really there is no effective treatment. It doesn't work. So as you can see here, this is a section of human eye. If we get the retina and magnify it, we can see that there are different cell types. There are photoreceptors and amacrine cells or horizontal cells. I'm not going to go to the detail, just the ganglion cell layers that the extremity of these ganglion cells can make the optic nerve that goes back to the brain, and we can see. But my focus is on these cells, which are called retinal pigment epithelium. And these cells are a monolayer of epithelial cells. While they look really simple, they have multiple function, and they are very important for health of the retina. So it works. Now, as I mentioned, RPE, or retinal pigment epithelium, are monolayer of epithelial cells. They have multiple function. They have the function of light absorption. They can do epithelial transport. They can transport various ions, and also they can regulate the 11 cis trans to 11 cis retinol, which is important for photoreceptors to capture the light. And also they have a very important role in phagocytosis of the outer segment of the photoreceptors, because the outer segment of photoreceptors is permanently shedding, and this is very important for renewal of photoreceptors. So if there is a problem at this point, then the photoreceptors get degenerated. They also secrete growth factors bipolarly, PEDF from the apical part and VEGF from the basolateral part. And they are sitting on a membrane which is called Bruch's membrane, and together with Bruch's membrane, RPE form the retinal blood barrier. So it's really important that RPE remain functional. And as I mentioned, there are two types of AMD. In the dry forms of AMD, they are deposits of drusens, which are lipids deposits. And depending on the size of these drusens, the physicians can diagnose whether or not the patients have AMD. And if once these deposits become bigger and bigger, they can break the membrane of these cells. And once the cells are no longer functional, so the nutrient and all the functions that RPE have to keep will be disrupted. In the wet form of AMD, there is neovascularization, formation of neovascularization, and the blood vessels sprouts throughout the retina, and it's called wet form because there is because of the presence of blood in the retina, and that induces also retinal degeneration. Now, as I mentioned, AMD is a multifactorial disease. So there are various factors that can contribute to AMD. First is age, of course, but other biological processes such as protein misfolding, mitochondrial metabolism, cholesterol and lipid metabolism, and phagocytosis and protein degradation can contribute to AMD. Reactive oxygen species, because light absorption is always, constantly we have light absorption that can induce and damage into the eye. And also environmental factors, smoking, diet, infection, lack of vitamin D, or many other factors can also contribute to AMD. And it's depending on the background of the individual and some reason that we don't know still, the patient can develop either dry or wet form. But it's important to mention that 90% of the cases is dry AMD and only 10% they develop wet AMD. And sometimes first the patient developed dry AMD, and in the later stages they developed wet AMD. So and it's also important to mention that for the wet form of AMD, there are some drugs such as anti-VEGF, anti-vascular endothelial growth factor, to inhibit the formation of new vessels. However, for dry AMD, apart from some high doses of antioxidant and vitamins, there is really no effective treatment available. I'm sure you all are familiar with induced pluripotent stem cells, for which Yamanaka got a Nobel Prize in 2010, I think. The reason I'm showing this is that really everybody is focusing on reprogram these cells to make induced pluripotent stem cells, and then these cells have the potential to be differentiated into various types of cells and then transplanted back to the patient. But what is really important, and the focus of my lab, is to use these disease cells if they have disease-relevant phenotype, to develop some kind of treatment to not only first understand the disease, but then develop treatments for AMD and do drug discovery. As you might know, there is a clinical trial in Japan today. It started, I think, last year in Riken Center for Developmental Biology, and they are using autologous patient-specific, of course, iPSC that they differentiate into RPE, and then they transplant these cells into the subretinal zone of patients underneath the RPE in order to regenerate the RPE that are degenerated. However, and there have been a paper published that the iPSC, once they are differentiated to RPE, they no longer induce tumor formation, and they are safe at least until 14 months in animals they have studied. So they started the clinical trial. However, recently there was a report showing that the clinical trials is stopped because of some mutations that they have found in the patient's drive iPSC-RPE. These mutations are either deleted genes or changes to the genes, including one in an oncogene. And that's a problem because it could induce tumor formation. And for that reason, in one patient, they stopped the study in order to do more research to see whether or not and the point is, they did not find these mutations in the primary cells that they got from the patients first. So these mutations are in iPSC. For that reason, for the safety issues, they stopped the clinical trial, for instance. Now, talking about that, I just want to mention that as I mentioned, my interest is in understanding the mechanism of disease, and I think the iPSC are great cells to do that. However, iPSC are cells that are really useful to study mechanism of disease for diseases that are caused by a single mutation. For example, there are papers showing the disease-relevant cellular phenotypes in, for example, neurons derived from cells of patients with familial form of Parkinson's disease, where there is a single mutation, or Alzheimer's disease. And they have been papers showing that there are disease-relevant cellular phenotypes. However, in multifactorial disease, it's more difficult to show these phenotypes. And that's the limitation. And that's why, for example, for AMD, it has been difficult, and for many other diseases such as AMD that are caused by multifactorial, So my lab has shown recently that we can differentiate these iPSC to RPE that show all specific markers of native RPE, and they also show gradually they show the expression of the genes that are specific genes in RPE. But more importantly, we showed that these cells are functional and mimic the function of native RPE. They have the same similar membrane potential as the native RPE. They have similar gene expression profile, and they are able to phagocytose the outer segment of photoreceptor, and they do secrete the vascular endothelial growth factor from the basal part of the cells. So that shows that because they mimic the function and phenotype of the native RPE, they are therefore good cells, probably for cell therapy, but also for disease modeling. However, we also found that we can see some abnormalities. First of all, not all cells have the same type of pigmentation. Some cells are not pigmented. We also could see a non-differentiated layer of cells, some overlapping cells, and tight junctions in multi-layer structure, and also a strange non-RPE cell. So although we have seen these problems, but it's possible, I believe, by selecting the right cells, just to expand the cells that have the right morphology, and by using also the scaffolds that are specific for RPE, we can overcome these limitations. So in order to study AMD, in my lab, what we did, we got the cells from the eyes of AMD donors, dead people who were organ donors. We got the eyes, we isolated the retina, the RPE cells, and as you can see here, we had two control, and they're all almost the same age, age match, except this patient, which was younger. So we have two control here, and we have two AMD. And we also got a sample from a patient with dry AMD. So these are, and I'm telling you why we did that. So we did first the single nucleotide polymorphism to see if there is any susceptibility loci here. And interestingly, in the control, we saw susceptibility loci, this doesn't work, for AMD here, whereas this patient, this donor never developed AMD. Oh, thank you. this donor never developed AMD and was normal. We also found a protective allele in one donor for AMD. However, this donor, because it was a heavy smoker, developed AMD. So that again shows the multifactorial basis of AMD, that not only, there are many factors, not only genetic or environmental, there are several factors that play a role. This patient is an 80-year-old female that is diagnosed clinically with AMD, AMD, and shows the susceptibility loci. So the reason we did this, because research have shown that the reprogramming might, first of all, affect the disease phenotypes of the cells. Also because the iPSC can retain the epigenetic memory of the tissue of origin. So to see if reprogramming could affect or if the tissue of origin is important, we got these eyes and then we got the patient cells too. So, and what we did is that we differentiated, we got the cells. So we had 4 RPE, 1 fibroblast culture. We cultured these cells, then we made the iPSC, induced pluripotent stem cells. As you can see here, we got colonies that are positive for TRA160, which is a marker, surface marker, then NANOG, and then this is DAPI. We also found that these cells, these colonies expressing the pluripotency factors, genes. Next step was to differentiate these cells into RPE. So here you can see a monolayer of RPE. We got these iPSCs, we put them in a defined media, and we differentiated them into retinal pigment epithelial cells. These are monolayer of retinal pigment epithelium cells, and we stained them with ZO1, which is a marker for zonula occludin, and it's a tight junction that is specific for epithelial cells and RPE. And as you can see, these cells have hexagonal morphology and express ZO1. The green fluorescence is for RPE65, which is a specific marker for RPE cells. And we also showed that we used the phagocytosis assay by purifying the outer segment of bovine photoreceptors and then conjugated with FITC, and then incubating these cells with that FITC conjugated photoreceptor. And as you can see, they do phagocytose the outer segment of photoreceptors, so they are functional. In addition, they show the gene expression of a specific gene expression of RPE, retinal pigment epithelium, compared to skin, which is negative, and also this is a control iPSC RPE. So we have generated all these cells, and then the next step was to compare between the control and the disease cells. And as you can see here, the disease cells have disintegrated mitochondria. Here is a higher magnification. As you can see, the membrane of mitochondria is totally damaged. Also, we have observed higher number of autophagosomes compared to normal. This is the skin. The iPSC RP derived from the skin of the patients. Again, we saw disintegrated mitochondria and autophagosomes. And here, another RPE from RPE. iPSC RPE derived from RPE, from the eye, shows also lipid accumulation in the cells compared to normal cells. So we saw some phenotypic problems. Then we cultured these cells in the presence of increased doses of H2O2 under oxidative stress to see how they behave, and this is a system that we actually set up in our lab. So after 48 hours incubation with different doses of H2O2, you can see that there is a distinct you can see that the normal and diseased are separated, and there is a significant difference between the susceptibility to oxidative stress-induced cell death between the normal and the diseased. All 3 diseased showed lower cell viability under the same concentration of H2O2. Interestingly, when we put these culture these cells for 5, 15, 30, and 1 hour under the oxidative stress, we also saw that there is higher level of reactive oxygen production in the disease cells compared to normal cells. So they are significantly different in terms of ROS production. And to confirm the phenotypic data that we saw, we measured the ATP production by these disease cells compared to normal. And as you can see here, the ATP production is significantly lower in disease cells compared to normal cells. Also, glycogen production was significantly higher in disease cells compared to normal cells. So, for all these reasons, we believe that iPSC-RPE from diseased, at least in one case of one patient and the case of the AMD donors, might not be a good source for transplantation because sooner or later they might develop the same disease phenotypes when we transplant them. And autologous cell therapy might not be a right way to go, other than taking in consideration the risk factors for tumorigenesis and all other safety issues. They might not be really reliable cells for transplantation based on the data we got. So, in summary, iPSC-RPE of AMD donors from RP of AMD donors exhibit diseased relevant cellular phenotypes. And interestingly, iPSC from skin fibroblasts of AMD patients also showed the disease phenotype, relevant cellular phenotype. And therefore, iPSC-RP from AMD patients might not be a reliable source for transplantation. However, these cells would be great cells for disease modeling and development of new therapies. So I would like to acknowledge people who work with me, Maria Kokonaki, my student, and my collaborator at Georgetown, my collaborator at NIH, and my collaborator at National Medical Center, and also Retinal Group of Washington, from whom, Dan Bernstein, from whom I get samples, patient samples, and also my grants from NIH and Bright Focus Foundation and Prevention of Blindness, and also Georgetown. Thank you for your attention. I will be glad to answer any questions.
Some questions?
Yes.
Can you give us some insights about the induction procedure?
For iPSC-RPE? Yeah, this is a well-known, Yamanaka, first of all, we got the Nobel Prize for that. So there are 4 genes that you can introduce into a finally differentiated cells, which are OCT4, NANOG, SOX2, and KLF4. Some people use combination of different genes. And then afterwards, you wait for 3 weeks, you see colonies, you pick them up, you put them in a defined condition, and then you have to do your controls to make sure they are pluripotent. And for differentiation, you have to put these cells in a suspension culture with inhibitors for TGF-beta and also activin, and you wait for 3-4 weeks until they become pigmented they become embryoid bodies that have pigmentations. And after 4 weeks, you put them in adherent culture and wait until they make a monolayer. And you have to wait, you have to be patient, really patient. You're welcome.
You reported some drawbacks in using donor patient-derived IBS. Do you think that if you use mesenchymal stem cells, you will get a better effect in regenerating macular degeneration? So you won't have to undergo all this complicated procedure and producing IPMs.
Yeah, if you... Yeah, instead of making iPSC, you can differentiate them. Yeah, you have to pay attention to one factor, and that is that iPSC behave like embryonic stem cells in a dish, means that they are pluripotent. But mesenchymal stem cells are multipotent, so they might not have the potential to be differentiated into RPE. They might become differentiated to mesenchymal lineage, but not to.
I think they're inducible cells. So if you put them in a particular conditioned medium so that they differentiate into this specific cell, I think they would. That's my opinion.
I'm not sure.
I don't think mesenchymal cells can go to this.
No.
They're not, right? We need endodermal lineage.
Yeah.
Mesenchymal cells can only go mesodermal lineage.
Yeah. Only mesodermal lineage. That's why iPSC are being used. Now, if you get retinal stem cells, that is the best way. But getting retinal stem cells from a adult patient, it's not easy to do. Yes.
Is there a difference in the VEGF isoform expression between dry and wet AMD?
During wet AMD, I think there is higher level of VEGF secretion, and that's why there is this anti-VEGF that is used. But in dry AMD, the VEGF is not a problem because there is no neovascularization.
It's the difference of the different isoforms of VEGF?
I don't know. I don't believe so. I just think that VEGF is over. It's there. If there is, I don't know, but I don't believe so. Yes.
Nice work indeed.
Thank you.
I want to ask about the density according to which you cultured your cells on transwells to get this monolayer with tight junction morphology or RP morphology. What densities have you tried, and what, according to your protocol, what is the best to obtain the highest yield of differentiated RP cells?
Yeah. The density actually is important. First, when you get the EBs, you have no control. You have to select the EBs that have the higher, that look more like RPE cells, or they have like these pigmented cells. Then you have to, depending on the transwell that you're using, depending on the surface of the transwell, you need to put sufficient cells. Now, I don't remember exactly the number of cells by heart, but you can only put, the optimum number of cells on a small transwell, which is less than a centimeter square. So, but that definitely, if you put even not, many cells.
Multi-layer adherence. That's my concern. I saw multi-layer adherence, so you would not be able to make sure that your cells are RP unless they are a very superficial layer. That's why if it's single layer, that's what my question. Does it make any difference that you have multi-layer and you have superficially differentiated cells, or you have only one layer and it's all differentiated?
No, usually it should be one single layer and all differentiated cells. We didn't see multi-layer of cells. Oh, you mean in the cells that I showed because that, as I mentioned, depending on, when you differentiate any type of cells from iPSCs, you cannot 100% control, all the feature of the cells. The only thing you can do is the best you can, reproduce the condition, in vitro condition that can give you the best morphology and the best differentiated cells. I don't think anybody can say 100% all the cells look really like neurons in the brain, or really, that's why, and you don't see that.
Because you spray them by the end before you're transplanting.
I'm not transplanting, first of all.
If you intend to do so, you would.
That's why you need to do electron microscopy imaging, you need to do FACS sorting, you need to do many, controls before doing this. But even Riken Institute, with doing all of these, then they figured out that there are mutations along the road, so they stopped that. So that's why I don't believe that much in cell therapy. I'm using them as a source for understanding the disease mechanism.
I believe that for wet AMD, one drug was FDA approved. It's Mucogen from iTech. It was about 7 years ago. Do you have any comment on that?
No.
wet AMD, there's also Lucentis.
Yeah, there are so many. I'm not working on wet
Yeah,
AMD.
there's many drugs.
I'm working on dry AMD. I'm interested in
dry is
dry
different.
AMD.
Yeah, well, I'm talking about wet AMD.
Yeah, there are drugs available for
Yeah,
wet
that's
AMD, but not all of them are very efficient neither
The
because
anti-
people
drugs
become
for
blind.
AMD are anti-VEGF. I think the
Yeah.
key question
It's
is.
Yeah.
anti-VEGF, yes.
Yeah.
This is mucogen, one of the most.
Very good. Is there any more questions? No, excellent. Well,
Thank you so much. Thank you. Just one thing.